Today we're going to talk about measurement, data, errors, and then in the second half of the webinar, Stephen will talk about binding curves and how to fit those. Interestingly enough, this is clearly not a biophysical technique, but in fact, the information and tips within this talk are actually applicable to all the techniques that have been covered during the lecture series. So in some ways, it's actually the most important talk to attend. What we're going to talk about today should be applied at all stages of any project. That includes the design of experiments, the actual physical measurement and use of a particular technique, and then post-data analysis. So in my part of the talk, I'm going to cover properties of measurement and data. This is obviously something that everyone here should really know already, but we're going to sort of cover it from a pretty basic level just to get everyone up to speed. So apologies if you already know this. I'm then going to make some comments about data fitting in general, and then some discussion about how you evaluate the goodness of fit within your data once you've finished this process. So why do we actually need to make measurements? Why do we need to collect data, and why do we need to then fit the data? So if you look at this quote from Lord Kelvin, you can see, I won't read it all out, but the nub of it is, when you cannot express your knowledge in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely in your thoughts advanced to the state of science, whatever the matter may be. This just reflects the fact that science is a subject that has to be understood universally by everyone in the same way. And therefore, the language that we need for science has to be universal. And that's the language of mathematics and relations between parameters expressed in mathematical conjugations. You can contrast that, for example, with an area such as art, where when you look at a work of art, everyone actually gets a different impression or a different experience from looking at that work of art. So then art is clearly an area where the experience is individual, art is for I, while science is all about we. So everyone has to understand the same thing. That's why we need to measure and fit our data. Interestingly enough, this Jackson Pollock painting, which is clearly art, has been analyzed scientifically in that there are large numbers of Jackson Pollock fake paintings out there. And it's been found that the original ones have elements of fractal geometry in them and can be identified analytically by a scientific analysis. Anyway, moving on then to why or what are the properties of measurement and data that we might be interested in. I'm sure you all know this already. Our data and our measurement process has elements which are accuracy, and this is how close a measurement reproduces the actual value of the parameter that we're trying to measure. In contrast to that, we have an element of precision. This is the difference that we see between multiple identical measurements, sometimes known as scatter in our data. So here we have two cases where our observations and our average of these observations give an accurate value near the center of the target. These are lower accuracy situations. And then here on the right, we have two cases of high precision, this one with low accuracy and this one with high accuracy. But in addition to accuracy and precision, we also need to think about an element of validity. That is how well the thing that we're measuring actually represents or reports on the property that we would like to examine and the property that we think our measurement is reporting on. And then finally, also measurement and data have an element of units. We need to be aware of that and just make sure we don't make any errors in considering the unit. So when we come to design and make measurements and collect our data, we would obviously like to be in this area of this plot where we have high accuracy and high precision. Perhaps the next best choice here is where we have accuracy, so the average, if we made sufficient measurements, is giving us a highly accurate value, but we have a reasonable amount of scatter, so we have fairly low precision. The next best case below that might be this situation here, where we have a very high level of precision, but all of the measurements we've made are not accurate. Just to point out that in this regime here, where we have a high precision of measurement, we can then actually rule out outliers for particular, if we make a series of measurements, whatever reason, one or more of them are outliers to the main data set, then we can actually remove those when we have high precision. This area here may be okay if you're interested in studying, for example, the difference between two cases, perhaps a mutant of a protein, and you're measuring some sort of property of the wild type and the mutant. If you make the assumption that the systematic lack of accuracy, this offset from the true value, is the same offset in both cases, then by comparing the two situations, you can work in this regime. And obviously we want to try and avoid having low precision and low accuracy. So how does it come about that we end up losing accuracy and precision? There are two sources of errors within our data, and we can divide them up into those which we could consider as random errors, sometimes known as noise, and systematic errors, sometimes known as mistakes. In the case of random errors, we have a variety of reasons. This is by no means a complete list, but for example, if we try and measure a very small signal on an instrument, we will end up with a lot of random noise in our data due to a low signal to noise. Similarly, if we used, for example, tools during the preparation of samples, such as pipettes or whatever, and the precision of these tools was low, then we would again end up with a high level of random noise. But also bear in mind that data itself and whatever we're studying may have stochastic noise within it, so that actually the number is not fixed or the value is not fixed, as well as any deterministic components within that measurement. So the key point though is that random errors, which are a lack of precision, will average out. So if you have these errors, you make enough measurements, they should average out. Systematic errors or mistakes, on the other hand, will represent a lack of accuracy, and these are not removed by averaging unless they are a one-off in the case of an outlier. So systematic errors, we might consider the instrument we're using isn't properly calibrated, we go off scale when we're trying to measure, so our signal is too large for the instrument to measure. Our pipettes may not be properly calibrated, similarly with pH meters, etc. Or the operator themselves may make a mistake. They may make an error in a calculation, preparing solution. They may forget to put something in an assay, etc., etc. This is not uncommon, and obviously this leads to a large systematic error. A couple of other points, though, is that if we're interested in measuring a property at a particular temperature, then we need to wait for our system to come to that temperature before we make the measurement, because the value of our parameter may change as a function of temperature. Similarly, if we have an equilibrium process, then we may need to wait a certain amount of time to reach the endpoint before we actually make the measurement. So the other properties of data and measurement are that they have units and validity. Obviously, all data has units, even if it's an arbitrary unit, such as volts or something, a signal on a detector. This can be related to other measurements and measurements on other occasions, providing the instrument is set up in the same way. And where people have used incorrect units or made errors in calculations, this can definitely be extremely costly. You only need to think about the Hubble telescope being one example, and then this Mars lander that they attempted to land on the surface of Mars. Somebody had used a deceleration value in an imperial unit rather than a metric unit, ending in the demise of the lander and costing large amounts of money. At a more basic level here in the LMB, things that we need to watch out for are the difference between joules and calories. And for example, when you use the gas constant in a calculation, make sure if you're combining it with other measurements that you're using the correct unit. Also be aware of the difference between log and natural log, a value of a factor 2.303, and then degrees centigrade and Kelvin. Another area is the validity of our measurement, the validity of our data to what we think we're going to try and observe. So beware of spurious correlations. Of course, you can make any correlation you like, such as the correlation between the drinking of wine and speaking French language, for example. This will give you a positive correlation. It doesn't necessarily mean that drinking wine enables you to speak French. At a more scientific level, in the basis of this talk, then if we look at this data here, where we have some sort of spectra recorded of a protein, and then the protein interacts with a ligand, and as a result of that, we get a change in the intensity, but we also get a change in the wavelength maxima of these spectra. So if we look at the intensity of the system at any wavelength, this will reflect the relative amounts of free and complex material. And Stephen will show you how you can use data such as this to get binding constants from the data. But the change in wavelength maxima, which would be the shift between this wavelength and this wavelength, is not a function of state, because there's a convolution of both intensity and wavelength maxima. So in this case, measuring the spectral maxima wavelength is not a valid way of looking at this equilibrium. Okay, so in the modern world, we're very blessed with having computers, and in that way, we're able to do nonlinear least squares data fitting relatively trivially. This is obviously not the case if you think back to your olden days, or if you're as old as I am, you will remember things like graph paper and so forth. However, there are some disadvantages of having this power in software that can do nonlinear least squares analysis. And there's a famous quote from a physicist, John von Neumann: if you give me 4 parameters, I can fit an elephant. And if you give me 5 parameters, I can make him wiggle his trunk. So the message to this is, if you put in enough parameters in your function when you're fitting, you can fit pretty well anything to some sort of mathematical function, including elephants. But then you have to ask yourself, is there still a model? Or am I just making this function go through the data that I collected? Interestingly enough, of course, this is quite a famous comment by this physicist. And so it led to a couple of attempts, which were published in the scientific literature, where people tried to recapitulate an elephant using different numbers of terms. And in this case, using 4 complex parameters, they were able to regenerate this picture of an elephant and make it wiggle its trunk, as you can see. So in all cases, it's best to use Occam's Razor, which is also known as the law of parsimony. And this can be just simply stated as the simplest explanation is usually the correct one. So what we would urge you to do, and what you should do, is to consider first the simplest model that you can that describes your data, and you would only invoke additional complexity if that's supported by other observations or perhaps some orthogonal data, and also that the fit to your simple model to the data is not particularly good. And I'll show you how to judge that later. So these are the kind of software programs that we use commonly in the LMB. Stephen and I use GraphPad Prism, and we're also fans of Kaleidograph. That's more of a historical link in that as you use software a lot, you become familiar with it and you're reluctant to change. There are other graphing packages such as Sigma Plot and Origin that are in the lab, and sometimes people use them. These all have very intuitive user interface. You'll see that later when Stephen looks at the binding data. You can normally enter the functions that you want as text, and you can have data nesting within plots, meaning that when you save the plot, you're able to access the data that you use to make that plot. And some of them allow you to do global fitting, where you fit multiple data sets to the same model. Excel obviously has a place, and it's sometimes quite good for simulation, although the plotting functions of Excel are not great. And then there are some more higher power mathematical packages which require programming, and there's usually a steep learning curve to engage with these, but certainly the sky's the limit if you use those packages. So when we do nonlinear least squares fitting, we have certain aims, and we're making certain assumptions about the data. So the aims we have are mainly to avoid having to transform our function into some sort of linear form, so that we can do simple regression on it. And again, I've said earlier, maybe some of you remember the old days with graph paper. So linearizations, which are very common, are the Lineweaver Burke plot and the scatter plot, etc. So by fitting the data in the raw form without transforming them or transforming the function to allow that fitting, there are certain advantages. And non-linear least squares tests if your given function is consistent with the data. This is the aim of it. It's not actually telling you whether your function is correct. It just determines the values of the parameters in the function that have the highest probability of getting as close to the data that you're giving the software to fit as possible. The assumptions that we're making when we do nonlinear least squares fitting are less commonly thought about, and perhaps can cause us some problems. So one of them is that we know the X value accurately. So if we're adding say we're varying the concentration of a ligand, we should really know that concentration very accurately when we've made our dilution series and the concentration of the stock. The only uncertainty in our data is in the dependent Y variable. The measurement points should be independent. Each measurement should be independent, which isn't the case if you do serial dilution. They should be uniformly distributed on the x-axis. The variability in Y is random and has Gaussian distribution, and the variability in Y is equal as a percentage for error for all points. If it isn't equal, then we may need to determine the individual errors on each Y point and fit the data using weighted errors, where we assign an individual error to each Y value. So what does the program actually do when we do the data fitting to our function? All the program's doing is trying to do least squares fit, and it makes small iterations in the variables which are present in your function, and it starts from values that you input at the beginning of the fitting. the guesses, if you like. And one thing to bear in mind is it may not find the logical and the global least squares minima unless you start fitting with reasonable values. So if you make your values wildly different from what they actually are, the program may find a local minima in this least squares surface and not actually converge on the best fit. The more variables you include in your function, in this case, we have 2 variables, so we have like a 3-dimensional fitting surface. The more variables you have, then the higher the dimensionality of this least squares fitting surface. And so the more local minima there are, and so your initial guesses may have to be better. Some programs usefully allow parameters to be constrained, meaning that you tell the software that this variable can only vary between an upper and a lower limit. This tends to constrain the fit within reasonable boundaries. And another approach you can use is to fix some of the parameters. So rather than having them as variables, you just input a number, and then you release those. Once you've then got the other variables fit, you then release the other parameters during a second stage of fitting. So how do we decide if our fit then that the program is giving us is any good? We advocate generally a much more common sense assessment of the quality of fit. Obviously, when you do a fit, you will get a series of values for your parameters, indices of the goodness of fit, the R value and the chi-squared. These do give some idea about how good the fits are. But it's important to bear in mind that the fit parameters that you obtain here and the errors on these fit parameters do not take any account of your measurement precision. So please do not present fitted values with these unwarranted levels of precision, all these decimal places, unless your measurement justifies that. So the questions we can ask in terms of common sense assessment of fit is, are the best fit parameters, are these values actually physically reasonable? Have we generated negative rates or negative concentrations, or perhaps a value which we judge to be physically unreasonable? The other thing is, do some of these parameters have insignificant values or very small amplitudes? So we might then consider that these are not necessary and we could take a step back and remove them from the fitting function. And similarly, we could ask ourselves, do any related parameters such as rate constants have similar values and so might be omitted in favor of a single term, again, applying Occams Razor. However, when you do this, you should bear in mind that if the data actually fit to a single rate constant, then this can mean a number of things in your experiment. Obviously, it's the justified thing to do based on a law of parsimony, but the conclusions you can draw if your data do fit to a single rate is that there is indeed only one rate. But however, there could also be 2 or more rates that have similar time constants and are not resolved as individual rates. There could also be 2 or more rates with all but one of them having very small signal change. So there may be numerous rates, but the actual measurement can't detect them. There's no amplitude associated with them. And there may also be 2 or more rates, all but one of them are faster or slower than the time window of your observation. So in terms of discriminating any of these alternatives, you then have to redesign your experiment and make further measurements. Another qualitative common sense assessment to fit is just to look at the data and look at the fit and ask yourself, is the fit any good? Does the fit go through all of the data points or all of my x over all values of x? And in the case of many measurements that are made, this may well require you to look at the data on a nonlinear scale, and a log view is particularly useful for biological processes such as kinetics or binding. And here you can see the linear representation of this kinetic trace, and the fit looks reasonably good, although you can't really see what's going on Here at very short times, when you look at the log of this plot, you can see the fit is extremely bad. So perhaps the most powerful way of assessing the quality of your fit is to look at something called the residuals. And the residuals are just simply the difference. That's the data, or sorry, the fit subtracted from the data, or you can do it the other way around if you like. The residuals are just simply the difference between the data and the fit at each data point. So you can see with that example, when we're looking at fitting this data to a single exponential function, the residuals are large and they vary in a systematic way. If we add in an additional exponential function, so we're now fitting this to a double exponential, you can see that the residuals then are randomly distributed around 0. So residuals should always be randomly and uniformly distributed in the case of a good fit. So finally, I'm just going to mention a couple of slides about statistics. When we come to the real world, we're interested in perhaps what is the error on our measurement. And the curve fitting process gives us errors, but these are just simply fitting errors. I've already made the point about the precision that they imply, the number of decimal places, but these fitting errors only report on the least squares fit to a particular data set. You may be able to reduce random errors in your determination by repeating measurements and making averaging, but systematic errors will not be removed by repeat measurements unless you remove the source of the systematic error. So taking care with calibration of instruments and pipettes, adjusting your protocol, giving enough time for systems to come to equilibrium. And ultimately, if you really want to know what the error is in your determination, you may need to repeat the measurement using fresh sources of all materials that are present in during your measurement to eliminate all sources of error. These are often known as biological replicates. So you would need to prepare all of your samples again and make the measurements again. And common sense again applies here, because the required precision, which you improve by going through these processes of repeating measurements, the required precision will dictate how far you need to progress along this route. So if you have 2 measurements and they differ by a very large amount, then obviously you can accept a larger error in those measurements and still be confident of the difference. Another comment is just that fitted values are sometimes combined or extrapolated, and if you do this, then that will compound the associated error. So there are simple formula that allow you to take account of the propagation of errors for the sum or difference and for the multiplication or division of parameters. And finally, just a word about the constraint or bias in data sets and an approaches to get around that. So if you remember, one of the assumptions of nonlinear least squares fitting is that the measurement points are independently and uniformly distributed. But sometimes when we collect data, as here's an example of some here, some of the points are more important in constraining the fit when the x-axis distribution is not uniform. So in this data set, we were keen to confirm that this data actually did have a negative slope on it. But there we were worried that these points, which are at this extreme of x-axis, were helping to generate this negative slope. And you quite commonly get a non-uniform distribution of data when you've transformed the data, such as in Lineweaver-Burke or Hill plots or whatever. So you can get around this to some extent by looking at the significance of these points using a statistical resampling method, and the common ones around are called bootstrapping or jackknifing. In the case of bootstrapping, which was applied to this data set, you generate a data set which takes the same number of data points as you have here, by randomly selecting them from the pool data set and then replacing the point after each selection. So once you've taken n, the same number of points into your synthetic data set, you fit that to get a slope and an intercept, and then you just repeat that process maybe 1,000 times. And you can see here, using this bootstrapping process, the slopes that we got on the data here using bootstrapping had this Gaussian distribution, and the mean of this distribution still had a negative slope, and so we were our confidence in that was increased. So now I'll hand over to Stephen, and Stephen will discuss and probably repeat some of the things that I've said, but there's no harm in that.
Okay, so I'll start with this quote from Richard Henderson, which is saying, one must never underestimate the ingenuity of humans to invent new ways to deceive themselves. And we'll see many examples of this in the next half an hour. So the first one is, here is a classic example. It's a functional MRI of an Atlantic salmon, which was showing pictures of human emotions. And look, hey, these brain regions kind of lit up here, and that was kind of showing, oh, right, so the salmon is actually reacting to these pictures. There was one problem. The salmon was dead. So this kind of just shows you an importance of backgrounds and making appropriate corrections. So, I mean, overall, I mean, the best thing to do, when you come to any of these experiments is to think first, and to think about, what's your biological model, what method and what controls you're going to use. Is it reproducible? And the reproducibility is really a bit of a crisis at the minute in science. I mean, pharma have found that many drug pipelines actually fail. And one of the reasons why they may fail is the underlying biology may be wrong. So Bayer Healthcare did a screen of 25% of published targets, and found that they could only reproduce 25% of them. Amgen did a similar study as well with 53 studies, and they could only find 11% of them could be confirmed. And then just before Christmas, there was a report from the Reproducibility Project, and specifically on cancer biology, where they tried to reproduce 112 different experiments, and they only had 46%. So you really need to ensure you kind of take note of all the controls and what we're going to say, not necessarily in fitting data, but also in your general scientific method. I mean, also journals are now, because of this problem in reproducibility, they are now expecting more clarity on how you've done experiments, and what your samples, how many reproductions you've done, whether they're biological or technical replicates. Okay, so how do we fit data? Well, you're going to have some kind of physical model. You're going to have to think, well, what signal do you have, and what data, what controls, how you're going to correct that data, what's the data model, and how you're going to fit that. So the question we'll look at in the next couple of minutes is that signal representative? What are the assumptions in there? Are your controls good enough? And as Chris mentioned just in the first half there, do you have the right data, and are you looking at it for the right time? So you come to a data model. What's the assumptions in that data model when you use that to fit the data? Which program are you going to use? Are you going to do global or single fitting? And how do these programs kind of show you the quality of the fit? And ultimately, of course, you're going to fit the data to the physical model, and that's going to inform the biological model as well. So let's go through this first bit then. Okay, here's an example of an enzyme that bound a substrate, and with fluorescence, we could see that when it bound ATP, there was a decrease in the fluorescence signal, and we wanted to find out what the binding constant was. So we could correct the data here, and this is against non-hydrolyzable ATP. As we increased it, then the decrease went down, it was saturated. And so then I'm going to fit this data. This is the data model here. And we're going to fit the fluorescence change, again, to get the KD. And the assumptions are then that it's a one-to-one binding, and that the concentration of the complex that's being formed is actually directly proportional to the change in fluorescence. And we also have to make the assumption that there's no further hydrolysis or turnover. And in this case, we were using a non-hydrolyzable ATP analogs. Of course, it may be that you've not got the right signal. Let me go back again. It could be that it's not a simple one-to-one binding. Your physical model is actually wrong, and there's all these other steps here. So what is this change in signal actually reporting on? So you always have to think, well, is there always missing steps, something else that's actually happening? And is the signal actually representative? Here's a case of looking at fluorescence polarization of a fluorescently labeled nucleotide analog. So this compound here actually bound into the kinase, and this is the fluorescence moiety here. So as you see, if you increase the kinase concentration, you get an increase in binding. That all looks pretty good. But what happened when we put the control in, just this fluorescein here, you saw exactly the same. So what was actually happening was the actual compound here, which is supposed to bind into the active site, wasn't binding in there. But what was happening was the fluorescent group was actually binding in there. So it was just non-specific. Okay, one way to kind of find, do I have a specific reaction would be to do displacement reactions. So here we've got a fluorescent reporter going in. And so can we titrate that out if we've got a known positive control, so we can compete out the fluorescent reporter and get down again. Here in green, it's going all the way down, blue, not so much. So as we've kind of mentioned all the way through these talks is that, good signals, need good samples. So make sure your buffers and your samples are all filtered or spun down. Make sure you don't have any aggregation before and also after the experiment. Are your concentrations accurate? How have you measured that? Is it just nanodrop on 280, or is it a Bradford? Is that accurate? Have you looked at your sample stability beforehand? We've got methods to do that. There could be changes in maybe the temperature, the pH, and the time as well. You've got to look at. So your methodology: have you got a good signal change? Have you got background, buffer effects, accurate titrations, temperature control, time to each equilibrium? Do you have good controls? Positive controls, negative controls? Are your reagents defined, and are they stable? So here's an example of getting a robust assay. This was a fluorescence polarization high throughput screen, again, for a kinase. Here's the reporter that was then binding onto the kinase. You get an increase in the polarization. And as you can see, as we kind of started to purify the protein and do the high throughput screens, we actually got better and better at making the protein and doing the assays. One way to kind of quantitate that is to actually compare what the signal window here. I should actually put on my laser pointer. And the signal window here against the variation in signal. And so that this is, to quantitate that is termed the set prime factor, which is then looking at the distribution of signals then against their actual overall values here to give you this set prime value. So a set prime value of 0.6, but 0.6 is really good, and you can see it's really robust. So that when we came to do the high throughput screen, if we wanted to look for compounds that then displaced this reporter, then we hopefully then get a signal in the middle here, and that would tell us we've got a hit. So this kind of shows you about technical replicates and also biological replicates. So these are different batches of protein. So these were biological replicates. And because this was a high throughput screen, then these controls were done across the hundreds of plates that were actually measured during each of these runs here. Okay, so when you set up your experiment, you've got valid controls, you've got offensive reagents, good targets. Then it's really, really important to have really good documentation to put down exactly what you've done, and to put that in your lab book and your thesis, so that not only the people who come after you in the lab will actually be able to reproduce it, but also when you write in papers as well, that other people may be able to reproduce your study. And it may be some part of that issue in reproducible studies where they couldn't reproduce things was maybe that things weren't particularly written up very well. Okay, another thing, of course, is to not waste time on using bad reagents. The classic quote here is don't waste clean thinking time on dirty enzymes. And here's a case in study that I did way back when I was looking at this chaperone HSP90, which is an ATP. It's got lots and lots of co-chaperones, and there was always a question at that time is, well, what do all these co-chaperones do? So I purified this one from E. coli called FKBP59. We put it into the ATPase assay. Here's HSP90. When we added in FKBP59, there was a massive boost in the very, very low rate of ATPase activity. So that was really exciting. Put it in with another co-chaperones, P23. Well, that inhibited the ATPase quite a bit. FKBP, wow, massive boost. And we put in the P23 as well. So we seemed to then suppress that. So that actually looked like there was something going on. It was really quite exciting until I purified the protein a bit further. So this was FKBP here, and this was a purification, further purification down, and it was an ion exchange column with a salt gradient, and I found I got two peaks. The first peak here was the FKBP59, and then there was a second peak here in fraction 39, which is round about here, and you couldn't really see, but when we put it into the assay, it was really quite revealing. First of all, here's HSP90, here's FKBP by itself, and here's fraction 39. So it's had a massive ATPase activity by itself. So even though I could hardly see it on the band here, this was just a contaminating factor. And really, subsequently when I put FKB59 with HSP90 in, there wasn't really any difference. So what's your background signal? You have to be really, really careful in terms of, say, your titrating your titratant is contributing to the signal. So it could be like light scattering, or in ITC, there's heated dilution. Here's an example here, which was just putting in DMSO. And what we could find, if you just put DMSO into the buffer, it was supposed to be matched, but actually it wasn't actually matched, and the DMSO was actually causing a signal just by itself, which you could actually fit. But obviously, it's just a change in heated dilution because the DMSO concentrations were mismatched. So when you have buffer mismatches, that's important also in SPR and also AUC and other things as well. Also, the titratant could actually affect the measurement itself. So if you say you're titrating in ATP and you're measuring the signal in the UV, the ATP itself will then absorb the light, and you'll get something that's called the inner filter effect. The fluorescence will go down as you increase the concentration of ATP. Okay, so as Chris was mentioning earlier on, you've got corrected data, but have you got the right time and the right data range? Here's a published example of a screen, and it's in using SPR. So the fragments are very small, so there's not very much of a change. But they all seem they fitted all the data to give you some, KDs. But, looking at them, you kind of think, well, I could fit this to really a linear function. Is this really right? So in the paper, they reference a methods in Genology Paper saying the shape of the binding curves can be uniquely defined by fixing the Rmax. So the K, and then you can get the KD. And so you can actually fit a curve where there's no inflection point. So, is that really reliable? Is that a real binding, or is it not? So as Chris mentioned earlier on, putting things into log is sometimes very revealing. So here's a binding curve, again, a fluorescence polarization. And have we actually got the full range? Well, if we have a look at the log view of concentration, we can see we've got a baseline here where there's the free fluorophore, and here we're getting up to saturation where there's no further change. But what happens if we don't have the right range of data? Imagine if we just take just the early phase here, and we don't do very many points. Okay, well, I can fit a curve, but let's have a look at the log view. Well, we're not actually getting up to saturation at all, and the KD that we're actually fitting is going to be higher than the final concentration of protein we are putting in. So if you ever get that, then alarm bells should sound, and you should think, right, I should go back and actually extend this into a higher concentration. Well, imagine if we only fit, not like in that previous example that was published on the SPR, where we haven't actually got even to the inflection point. Well, too short a range, we can still fit. Maybe we're fitting because this is just stochastic kind of variation. Because we've only got a few points here, we may be able to fit a curve, but is it right? Well, let's have a look at the log view. And again, we've got a very, very small amplitude here than we would expect for binding this protein. And the KD that we're going to get is actually going to be much tighter than it is in reality. So the rule of thumb would be for a studio titration to the final concentration is at least 5 to 20 times above your KD. So it's really important that you actually go to saturation, fit the raw data, and not do what some people would do, say, well, I'm definitely at saturation here. I'm just going to normalize the curve. It's very easy to normalize. Yeah, that looks like a nice binding curve. But if we look at the real data set, in blue here, we can see that we really underestimated where the KD is. So am I looking at the right view? Let's have a look at the long view. So you can imagine that each individual binding constant here is the result of the underlying intrinsic on and off rates of this binding. So that the overall rate is going to depend upon these, and when you get to equilibrium, they're going to be equal. And here's an extreme example here where equilibrium wasn't reached. So you could actually fit a binding curve, but actually as you incubate it over time, you can see the amplitude was going up and also the KD was going down. So it's always important, especially in, say, if you're doing plate-based assays where it's very easy just to set up the whole plate and then stick it in, measure it, and go, well, there we go, there's my result. With a plate, you can incubate it for half an hour, 2 hours, overnight. Obviously, it's useful to then put a cover over that to stop evaporation, and then compare all those different signals at different times and say, well, are you getting the same binding constant, or is there something subtly shifting because you've got something slow kinetics underlying the binding constant? So are you taking the right time range? If you're looking at kinetics, have you reached that endpoint? So here, if you say we don't do a very limited range, and we've kind of mentioned that in the SPR, where sometimes you can't really get to that endpoint because the kinetics are just so slow. So the danger in that is that, well, you can fit a curve, but you might just end up with the completely wrong amplitude and the wrong rate. So the rule of thumb there would be, if you want to get to the point, where you've kind of reached the baseline, then you need to go from 10 to 20 times the half time. Conversely, when you're doing enzyme kinetics, if you want to kind of generate a Kitten curve, you have to make the assumption in this data model that there is no product formed. So therefore, you must look at the initial rates. You must have the enzyme at a concentration way, way below the substrate concentration, so that in this x-axis, that the substrate you put in is essentially the same as the free substrate. And again, to make sure that there's very, very little product, which is essentially 0, you need to look at the initial rates to fit those. So appropriate concentrations when you're doing titrations. Well, if you say if you're doing fluorescence in MST, usually what you want to have is the total label component below or at least equal to the KD. And you have to make the assumption then if you're that the titrating peak doesn't change that much. If it does, then if your concentration of this labeled material is quite close to the KD, then you need to use a model that will actually take into account what's termed the ligand depletion during the assay. When you fit data and you end up with a KD that ends up being about half the concentration of the total ligand concentration, then again, alarm bells must sound because your affinity may just be very, very tight. And so that what you're really doing is you're just fitting, you're just titrating stoichiometrically. So go down, then you need to go back and think about that. Obviously, in ITC, it's the other way around. The ligand concentration needs to be in the cell needs to be bigger than the KD, so you can titrate. Obviously, when you start off with these kind of experiments, you don't know what the KD is, so you just have to have a rough idea, maybe figure it out roughly, and then go back to measure to get accurate concentrations. Okay, so you've got lovely data. You've ticked all these boxes here. So let's have a look at data fitting. So one thing, what assumptions have you made in that data model? Do all the parameter rates of the signal. it's easy, very, very easy to get, a good fit with bad data. Here's an example here. There's only, 5 or 6 points here. The person used Prism one-site binding, which has got this non-specific binding linear phase here to the binding. You could fit it, but it's completely wrong. So you have to say, well, why do I need this kind of non-specific term? Here's some published data, which I would say is like, it's the wrong shoe and in the wrong feet, and you get lots of pinches and gaps here in the data. So this is the squiggly lines are obviously the data, and here's the solid lines are the fits. You don't really need to kind of see what the residuals are to know that this isn't fitting very well. So they assumed it was one-to-one interaction, and they actually quoted kinetics to 2 decimal places because that's what the program outputs in terms of the fitting. So sometimes when we kind of think about is all the data good? Sometimes you have to remove outliers. But what's the difference? But how do you judge what's an outlier and what's outright lying? So in ITC, you always get this initial point here, which is you have to take out. Now we need to do that because when you have the titrate in the syringe and you put it into the cell, then sometimes you might get a bit of diffusion from the cell into the syringe, and vice versa. So we always just titrate a very, very small volume just to get rid of that sample, and then we have to remove it in the data to actually be able to fit. So here's another case where you kind of go, well, I've looked like I've reached saturation, but there's something happening up here at the top. Oh, there's bad points. Let me try just take those out. But of course, you need to think about, well, why am I taking this out? You're getting a better fit. Is there something like aggregation happening? But if there's aggregation happening here, is there aggregation happening here? So where does that start? You may be just cheating because, say, we extend the titration and you find actually there's a second step here. So that these are actually real data steps. So as Chris was saying, look at the variation. Is it Gaussian? Can you think of why is this experimental error maybe at the top here? Can you get better assay conditions if it was segregation? Do you have the wrong data or the wrong actual model itself? Right. So in the last kind of few minutes, we'll just talk about what data programs look like. So here's Kaleidograph. Me and Chris both like it because not only it's very simple to use. You can just put your data into columns. It's got a very easy transformation tool. So if say you've got the wrong units or whatever, then you can easily transform that. So you just get a plot, and of course we're always fitting raw data, not normalized data. It's also got a range of fitting programs in there. You can also write your own, and you can save the models. And so here's a data model here. I won't go for the derivation. Go back to the fluorescence spectroscopy lecture I gave, and you can go through this. But essentially, this is the data model here. And in Kaleidograph, then we can put this data model into these parameters here, which is called the M. We can input the values that we know. In this case, this is the total ligand concentration. We can put that in because we know what it is, using the right units for what we use the protein concentration as. And this is M naught is the x-axis. So we put that in, and then we'll get fitting out here. So this is the values and the errors and the R squared, the chi-squared here and the R squared. But you can see the value of fit. And we can look at the M and the x-axis, which is the log view. So here's another data model where we've made the assumption then that just the change in the fluorescence is directly proportional to the concentration of complex formed. And so this is a much simpler equation here. And we've also put in estimates here to we can kind of judge, well, this is what our amplitude is, and we think the KD is around about here, so we can fit. But what if we put in the wrong estimates? Well, it'll still fit. And wow, look at that R value, that's really, really good. But then, look at the chi-squared and then look at the errors here. So obviously you always need kind of good estimates. So here in Prism, again, data is in columns. We can fit that with a nonlinear regression. And again, there's either, they've got a whole list of equations already in the program, but it's always better to write your own. It's a bit easier to write them in Prism because you can put in words and you can define what they are, and you can define constraints and put initial rules here. So you can kind of say here the bottom is going to be the Y min, Y max. This is going to be the concentration of the protein which we need to put in, and we can put in a value for KD. We can also after constrain these, like say, well, this is going to be a constant. This is the ligand concentration, so we need to put that in there. And we can say, well, okay, the binding constant, well, that's definitely going to be above zero. We can put initial values in here. There's automatic ones, but we can also put in ones of our own. And then we'll get out a fitting like this. And again, this is going to be the values and also the standard error of the fit. I give you confidence levels. So, where this value, say, of the top will compete between. And so if it's quite narrow, then that, okay, that's well constrained. And this gives you the R squared here of how good the fit is to the data. If you want to look at, say, residuals that Chris talked about earlier on, in Kaleidograph, we just view results, and then we can copy the residuals to a data window and fit. In Prism, there's a whole diagnostic window, or you could put the SE, and also put down here, run a residual plot to make these. And again, when we're looking at the residuals, we're looking for the simplest one, which will actually fit the data and not give a kind of a systematic deviation. Okay, so some programs for different instruments will generate residuals by themselves. Here's SPR, here's the residuals. And let's come on to then just how do you fit? Do you fit individually, or do you fit globally? What do you do? Well, I mean, sometimes if you have to constrain all the data to fit one set of parameters, and that might be because it's very difficult to fit in, like, say, AUC. But you always have to be aware that are you just doing a global fit to get data fit because you've actually got bad data. So it's always good to kind of think about, well, look at all the individual parameters across all the different concentrations, because you're only going to, and for some parameters, you're only going to get one shared parameter, but you can have a look at the amplitudes and the variation in other ones. In Prism, then you can kind of say, well, I'm going to, for the K on and the K off, you can say share for all the data sets, and then the other ones are maybe not constrained. So you may look at the data and say, well, actually, maybe the amplitude on, say, some of these low concentration isn't very good. A good thing to do is maybe take that data out and see how that data varies, how the fits actually vary. And again, similarly, if you're high concentrations, well, maybe something else is starting to happen here. Maybe you're starting to get non-specific binding, and so your data model isn't actually fitting data at that high concentration. So here's an example of, say, with SPR, fitting globally. Can we compare this? If we fit the kinetics against the equilibrium response, and so if you fit globally, you get 1 point, and then you can compare that against fitting singly. Do they correlate? And here, and the statistics, okay, it looks like maybe, okay, this on rate, it's not really, it doesn't really representing the average of the on rates if you fit individually. Why is that? So you need to go and have a look at that. Sometimes you can get it wrong by globally fitting. I mentioned this in the SPR lecture, where fitting on off rate wasn't globally off rate, gave very, very poor fitting values. So, fitting globally gives us a very, very tight KD. Fitting individually gives us a substantially different KD, which was similar to the ITC. And that emphasizes that if you want to know whether your physical signals and your different physical signals give the same answer, do different techniques and compare them, say, like fluorescence, polarization, or ITC. And of course, the thing is to repeat, and repeat. So lastly, again, you can just say that in Kaleidograph, you can do statistics in columns, and you can plot those as these kind of dot plots. And in Prism as well, then you can do then column statistics on all your values to get these kind of plots, see the variation. And you could decide, what kind of parameters you're going to plot on there. Of course, you can do P analysis on this to see significance, but there's in the literature, there's obviously a lot of controversy about P values. Very careful about that, but that's a bit. There's not much time to talk about that in this lecture. And what does this KD actually really mean physically? Well, it relates to then the energy of the interaction. So how much of a change of KD is really significant. Okay, so in the end, you fitted your data. What does it all mean? Hopefully the physical model is going to tell you something about the biology. So as Chris said, Occams Razor, choose the simplest model, but there's another view as well, which was first kind of said by Sidney Brenner, who's leader of this parish, who said, well, maybe if there's lots of, say, errors and system, errors coming through, and you're kind of disregarding them, and maybe your physical model isn't necessarily matching the data, then perhaps maybe there's something fundamentally wrong in how people think that the biology is actually working. So maybe sometimes you have to lift up the carpet and actually sweep underneath it and actually see, well, maybe there is something in those experiments that go wrong. Maybe the biology is different. So thanks for you all kind of coming and staying with us.